Today in What's New in 22q, we're diving deep into, well, a really fascinating piece of the 22q11 deletion syndrome puzzle. It's research that might just shift how we understand and maybe approach some of its challenges. We're looking at a study that reveals this kind of hidden link between cellular energy, the brain's own defenses, and, yeah, crucial social behaviors. And it points towards maybe an unexpected helper, a medication that's already out there. So we're focusing on a recent deep dive published by Alexis M. Crockett and colleagues, specifically the main authors Stuart A. Anderson and Jorge Even Alvarez. It's in the journal Science Translational Medicine, published in 2025. And the title is Bezafibrate Improves Mitochondrial Function, Blood-Brain Barrier Integrity, and Social Deficits in Models of 22q11.2 Deletion Syndrome. Our mission really for this deep dive is to unpack this important study for you, to help you understand how a medicine already used for other things might offer some new hope for 22q11.
Yeah, and what's truly fascinating here is how this research shows this fundamental connection, like almost a domino effect, you could say, between how our cells make energy, the brain's protective systems, and then ultimately these complex behaviors, all in the context of 22q11.2 deletion syndrome models. It's really a multi-layered discovery tying these things together.
Okay, so let's break this down a bit. For anyone who might be new to what's new in 22q, 22q11.2 deletion syndrome, or 22QDS as it's often shortened, it's a genetic condition, and it can increase the risk for various health challenges, including neuropsychiatric issues, things like learning differences, ADHD, even a significantly increased risk for conditions like autism spectrum disorders and schizophrenia. It's complex.
Yeah.
So every bit of new understanding really helps us move towards better support.
Exactly. And to really get to the heart of this particular study, we need to start with the brain's, well, its incredible protective shield, the blood-brain barrier, the BBB. Think of it like the brain's really selective, highly trained security team. Okay, and it's not just a wall, it's made of specialized cells that carefully control what gets in and out of the brain, protecting it from potentially harmful things in the blood, but letting the good stuff, the nutrients through. Its proper functioning is just absolutely critical for brain health, keeping everything stable and safe in there. Right, so it's not just a passive fence, it's more like a really active border control with rules. And if that team isn't working right, the brain could be vulnerable. Precisely. If that barrier gets compromised, yeah, it can open the door for problems.
And we know from other research, don't we, that problems with the BBB have been linked to, well, a whole range of neurological conditions, things sometimes associated with 22QDS like autism or schizophrenia, but also things like Parkinson's. So finding an issue here in 22QDS models, that would be a pretty big deal.
It would indeed.
And what's really compelling is how this study connects those dots. It builds on something researchers have suspected for a while in 22QDS. See, if you follow 22QDS research, you'll know the deletion actually includes 6 genes involved in mitochondrial function. Mitochondria, they're the powerhouses, right? Generating energy for our cells. So there's long been this suspicion that maybe these cellular energy issues are a key factor in 22QDS. So this study asks a really crucial kind of cascading question. Do these mitochondrial problems specifically hit those specialized cells of the blood-brain barrier in 22QDS? And if they do, how does that affect brain function and, maybe most importantly, behavior? It's connecting the tiny cell stuff to the bigger picture.
Right. So to investigate this, the researchers used 2 main models. First, human brain microvascular endothelial cells, basically the cells that make up the BBB grown in the lab. These were derived from people with 22QDS. And second, they used a special mouse model of 22QDS, one that has a similar genetic deletion. And what they found was pretty clear evidence of mitochondrial impairment, problems with the mitochondria in the BBB cells, from both the human cells and the mouse models. Essentially, the energy engines in these really critical barrier cells, they just weren't working efficiently. They weren't producing enough energy.
Okay, so the power generators were faulty in the security team's headquarters.
That's a good way to put it. And this energy deficit, this lack of power, then directly correlated with a weakening of the BBB's protective function. It basically became more leaky, less effective at guarding the brain. And this leakiness, they saw it both in the lab dishes and, importantly, in the mouse models, especially in the hippocampus.
The hippocampus, that's crucial for memory, right? And social behavior, too.
Exactly. Memory and social behavior. Key areas often impacted in neurodevelopmental conditions.
So we've got this core problem identified, a leaky brain barrier, and it seems to be linked to these faulty cellular power generators. The next logical step then is, well, can we fix it? Did they look for something new or?
Well, and this is what makes the study so promising. They turned to a compound called bezafibrate. Now, this isn't some brand new experimental drug. It's actually a medication that's already approved and used for other metabolic conditions. And it's known to stimulate mitochondrial biogenesis, which in simple terms means it helps mitochondria work better, more efficiently, like giving those cellular engines a tune-up, basically, to boost their energy output.
That's a huge potential advantage, isn't it, using an existing drug? So what happened when they gave it to the models? Did it work?
The results were quite striking. When they treated the 22q-DS cells in the mouse models with bezafibrate, they saw remarkable improvements. In the lab-grown human cells, the bezafibrate improved mitochondrial function, yes, but it also strengthened the BBB's integrity. They showed this by better barrier function overall and increased expression of important proteins like claudin-5. Think of claudin-5 like the molecular zippers or maybe tight junctions that seal the gaps between the BBB cells. Bezafibrate helped make those seals tighter.
So it helps zip up the leaky barrier?
Precisely, making it less permeable, more robust. And in the mouse model, just 4 weeks of treatment with bezafibrate also improved the mitochondrial health in those BBB cells. And importantly, it significantly decreased the leakiness of the barrier, specifically in the hippocampus.
Wow. Okay, that's impressive.
And what's really fascinating here is that even though bezafibrate didn't seem to get into the brain itself very well, it showed poor penetration, it still had this strong positive effect on the BBB endothelial cells, which strongly suggests it's having a direct therapeutic effect on the barrier itself.
Huh. Almost like it's a barrier whisperer. It doesn't need to get inside the house, so to speak, to fix the front door. That's quite elegant.
It is, isn't it? A highly targeted effect, potentially.
So let me see if I've got this right. The core idea is, in these 22q-DS models, there's a genetic vulnerability affecting energy production that leads to a leaky brain barrier.
Yes.
And fixing that leak with this existing drug actually improves social memory in the mice. That's the chain reaction.
That's the chain reaction they observed. And connecting it back to families, this is perhaps the most exciting part. That improvement in BBB integrity in the mice was associated with an improvement in social memory. Now, social memory is a type of behavior we know can be affected in 22q-DS, and it's linked to that hippocampal function we mentioned. For families, this is potentially huge because challenges with social interaction and understanding can be so central. To think we might be able to influence this, even indirectly, at a cellular level by fixing the barrier, well, that's truly exciting. The study actually found a direct correlation. The less leaky the BBB became after treatment, the better the social memory performance in those mice. It really suggests that strengthening the BBB might directly help with social cognition.
That's a powerful connection.
And importantly, the bezafibrate treatment didn't seem to affect other behaviors in the mice, like hyperactivity or anxiety, or other memory tasks that weren't specifically linked to BBB integrity in this context. That specificity is really important. It helps suggest they're hitting a particular mechanism, not just having some broad, general effect.
Okay, so this feels like a genuinely hopeful area of research, but what does this all mean for families living with 22q-DS right now and, maybe looking towards the future?
Yeah, that's the crucial question, isn't it? It's really important to stress, first off, that bezafibrate isn't currently prescribed for 22q-DS. This is still research. But the research itself is incredibly significant. For families, I think it offers a clearer, maybe more tangible understanding of some of the biology that might be contributing to neuropsychiatric challenges. It highlights that these problems with cellular energy production, specifically within the brain's protective barrier, could be a really key factor. It helps us move maybe beyond just observing symptoms towards understanding some potential root causes. And that knowledge itself can be empowering.
It really helps connect the dots, doesn't it, between the genetics, the cells, the brain structure, and then behavior. And does knowing this maybe open up other avenues? Like, could lifestyle things that support cell health potentially help the BBB too, even while we wait for more drug research?
That's an excellent point. While this study focused on bezafibrate, it certainly reinforces the general importance of things that support overall cellular health. Good nutrition, adequate sleep, managing stress, things that are good for brain health generally. This research doesn't give us specific new non-drug treatments today, but it absolutely gives us a more informed perspective on what might be happening at a fundamental biological level. And looking forward, the fact that bezafibrate is an existing, approved medication for other things, well, that's a massive advantage. If, and it's still an if, future human clinical trials were to be successful for 22q-DS, the path from this kind of promising research to an actual accessible treatment could potentially be much, much faster than developing a totally new drug from the ground up. So that offers a very positive outlook for future therapeutic development. Real hope for targeted interventions down the line.
That speed to market potential is huge, definitely. So, okay, while it's still in the research phase, using models, it does provide genuine hope. Hope for very specific, targeted interventions maybe sometime in the future. It's not a magic bullet, but it points to a mechanism we might be able to influence. It's just another reminder, really, that by digging into these tiny, intricate biological details, we can sometimes unlock solutions for really complex challenges. And this deep dive has been brought to you by 22q11 Europe, the European Association supporting families living with 22q11 Deletion Syndrome. This research really highlights how even the tiniest parts of our cells, like mitochondria and these crucial structures like the blood-brain barrier, can have such a profound ripple effect, affecting complex things like how we interact socially, our overall brain health. It really makes you wonder, doesn't it, what other subtle cellular mechanisms are playing out in our health that we just haven't fully grasped yet, and how future research, maybe sparked by insights like this one, might uncover more surprising connections, opening up new ways to help and support people with many different conditions. Thank you for listening to What's New in 22Q, produced by Dr. Stefan Elias for 22Q11 Europe. Take care and talk to you soon.